today, and we have heard a lot of great talks today about technologies, tools, models, artificial intelligence, and I'm sure that I cannot add anything to these topics, but I really hope that I can add with a little bit of a clinical perspective on what we hope from lifetime to achieve for the field of cancer precision medicine. And this, of course, needs a start where we are today and also what kind of information we are lacking with our current approaches so far. So we are in Berlin, so we have to start with Rudolf Virchow. Genevieve already mentioned it this morning. So it was here in Berlin where Rudolf Virchow worked at the Charité and where he published his important work, Cellular Pathology, and he stated his famous words, omnis cellula et cellula, every cell is derived from a cell. So this is more than 160 years ago. Today, 2019, we are here with Lifetime, and we state the interpreters of the Book of Life are our cells. So it's either still important or we haven't learned anything in this focus. I guess it is still important, and what we have learned so far and what gives us new tools in our hands now are the new technologies. While Rudolf Virchow had to work with a microscope only, he could put the cells into focus, but he could not get a better view on the cells and their meaning. So the goals of Lifetime to understand how genomes function within cells, how cells form tissues, and how cells dynamically remodel their activities in disease progression was not feasible for him, but it is feasible today. So what about the role of single cells in cancer research? There is a nice review in Nature Reviews Cancer in 2017 by Beslan and Hicks, and they stated it in a more global view. The fundamental operative unit of a cancer is the genetically and epigenetically innovative single cell. Single-cell analysis provide the ultimate level of resolution in our quest for a fundamental understanding of this disease. This is certainly true, but of course we also have to get into some more details here. So what today is the workflow of single-cell analysis in cancer? Of course we have to spend some time to look for the biomaterial and to collect the right biomaterial from the different cancer locations. This is the primary tumor, but of course also the metastasis, and today the blood or the bone marrow with circulating tumor cells. And then something very essential is coming up. We have to isolate the single cancer cells, and ideally we need fresh tissue. This is important from the clinician point of view because this is not our routine way of dealing with tissue in the operating theater. We are used to collect material, send it to pathology, and this is paraffin embedded. With the more research-interested clinical departments, it's also feasible to store snap-frozen material, and some of the single-cell applications, of course, can be done with this material. But if you really want to have fresh material, this needs new logistics that we have to build up and establish so that we can collect this material for the analysis. And then, of course, you know better than me, there are different isolation methods for the cells, and there are different technologies for single-cell sequencing around that, of course, are also applied in the cancer field. So let's have a look where we are in cancer precision medicine today. So this is a high-risk cancer patient, and digesting your talk, I apologize that even me as a woman chose a male figure to represent the cancer patient and not a female figure. So what do we do today? We take a single biopsy out of the primary tumor at some location of the primary tumor. We take this tumor biopsy. We do bulk tumor sequencing on different technological omics platforms. We get a sequencing report. Then, from my perspective, some bioinformatics voodoo is done. We have defined an algorithm prioritizing the targets out of the data, and then we make a choice of a targeted drug, which we like to believe is a magic drug that will cure our patient. So how does this work in routine clinical life? I'm a pediatric oncologist, so my examples most of the time are derived from pediatric oncology. This is an initiative that colleagues at the DKFZ in Heidelberg launched a couple of years ago. It was the first personalized pediatric cancer medicine trial for relapsed pediatric cancer cases in Germany. And this shows how complicated such a logistics network has to be set up. So 58 German centers treating children with cancer hooked up and sent in their biomaterial to be profiled on a multi-omics platform. And after a while, it was feasible within 21 days to obtain a final report on potential molecular targets to choose from molecular targeted therapies. It was a great achievement to have an atlas of targets that are around in the major pediatric cancer entities in these relapsed patients. And of course, we want to make use of this data to treat the patients more successfully in a situation where the cure rates are usually less than 10%. So there were some great achievements. We cured a few of the patients by selecting molecular targeted drugs. But for the majority of these patients, we have to state that it didn't work so well because either they didn't respond or they had a development of secondary resistance, and after a couple of weeks with molecular targeted treatment, it didn't work anymore. So what's the problem with this kind of analysis? So what we probably do if we do bulk sequencing from a single biopsy, we identify the dominant clone or an average clone, and then we base every kind of drug selection on this clone, but we might miss more rare subclones in the cancer that we have not identified by this bulk sequencing approach. And these are probably the clones that come up again after we have eradicated the one drug that we already identified. So if you think about this limitation, it all comes up to tumor heterogeneity, and in the majority of cases, we tend to ignore it. Tumor heterogeneity is not only there between patients, it's there between a single tumor, but it's also there between the tumor and the single metastasis of the tumor, and it's even there intrametastatically. So we have a complex picture here that we can only solve with single-cell resolution. This is just a quick example of our own data. We tried to assess it in real patient material. So from 10 primary neuroblastoma patients, we did biopsies from 5 different locations in the same primary tumor, and we didn't do single-cell sequencing, but multi-region sequencing in this case, but still, we came up with a huge number of mutations that were specific in one of the 5 locations. So there is a huge amount of heterogeneity, up to 35%. And this is also true for highly cancer-relevant genes. So it's an important finding, and we have to evaluate what this means for the clinical arena. So there are certain limitations of current approaches that we hope to address with lifetime. One of it is tumor heterogeneity. We cannot calculate mutation rates if we only do bulk sequencing. We don't find the rare cancer stem cells in dominant other clones. We can't identify subpopulations of cells that are the ones that invade and then finally lead to metastasis, and this is our clinical problem. We cannot say anything about plasticity of cancer cells, so there is something called the epithelial mesenchymal transition, which is also an important dissemination step. We don't know anything about it if we just characterize bulk sequencing samples. Circulating tumor cells is another issue. These guys are very rare in the blood or in the bone marrow, and with bulk sequencing technologies, the sensitivity is not high enough to pick up the cells, let alone to characterize them. We cannot learn about clonal evolution over time, and we also cannot monitor therapy response if we only focus on the bulk tumor cells. I like this picture from the company QIAGEN because it makes also for lay people very understandable what the difference is. So if we regard the identification of cell subpopulations, doing this with bulk results, it's kind of like a smoothie. But single-cell technologies give us the opportunity to really see the fruit salad behind it. In respect to the detection and analysis of rare cells, it's even better, so we can identify and separate the strawberries from the bananas and the other fruits, while with the bulk result, we still have the smoothie and we can't look into it more differentially. So we need high single-cell resolution in the cancer field to understand the couple of features that the cancer cells bring about. First of all, we want to reliably identify drug targets. For this, we need to precisely describe all the cell population, also including cancer stem cells, in the tumor, but also in its microenvironment. The second point is we want to understand cancer pathogenesis to obtain evidence for models of cancer origin and progression. We would like to understand cancer evolution by inferring timing of mutations and drivers. We would like to understand metastasis because this is clinically the highest challenge. We need to define the cell populations that are responsible for invasion, for dissemination, and for homing in different organs. We need to understand plasticity, defining the features of epithelial mesenchymal transition, but also other phenotypic cell states that are maybe only modeled by epigenetic features and not by genetics. And finally, our goal is to understand resistance, evaluating and monitoring the therapy response of our patients. Let me give you some more detail in each of these fields and also come up with an example where we are today. So the first point, identify drug targets. This is what we today do with fresh or snap-frozen tumor tissue, in general at the time of diagnosis. The first technologies used were single-cell RNA-seq, which is able to resolve each clone within a tumor, identifies rare populations, and shows frequency of potential drug targets, if it's just a minor population or maybe ubiquitous expression. We have to consider that mutations and fusion genes today are the major drug targets, so we also need to learn precisely about them. This means a combination of single-cell RNA-seq and genomic DNA and cDNA genotyping is needed if we want to get the full picture in the cancer field. There's been a nice report on a new combination technique called target-seq this year, published in Molecular Cell, and I really like this approach to get more precision into these kind of technology combinations. And of course, multi-omics approaches are evolving. For example, also incorporating mass cytometry information. We may not forget the complex cancer ecosystem in this way, because it's not only the tumor cells, but it's the tumor microenvironment, a heterogeneous population of cells, including malignant cells, stromal cells, immune cells, vascular cells. So if we do this only with bulk sequencing, we get an average signal of thousands of cells with unknown composition. With single-cell sequencing, we have the power to dissect the cellular composition. And of course, the next level will be to incorporate spatial information into this complex picture. Just to have a look into two examples how this can be applied in the cancer field. This is a nice story published in Science in 2016, dissecting the multicellular ecosystem of metastatic melanoma by single-cell RNA-seq. And the first studies, of course, were rather descriptive, but they could tell us about the different intra-tumor heterogeneity aspects. This was, in this study, associated with the cell cycle, as expected, with the spatial context. But also, these early trials were able to identify, for example, drug resistance programs or a T-cell exhaustion program, which is important for immune escape of tumors. Then it got more sophisticated, so this is another nice example that is focused on the innate immune landscape in early lung adenocarcinoma by paired single-cell analysis. And here we find a combination of barcoding and mass cytometry. So this is a simultaneous single-cell analysis of tumor, of healthy lung, and of peripheral blood. And the authors come up with a unique tumor immune signature in lung adenocarcinoma and with a detailed immune cell atlas of the human lung. And both, of course, is important to design more efficient immunotherapies. So this is creating the basis for our future treatment approaches. The second part is on understanding cancer pathogenesis. And here I would like to share a beautiful example that has only been published last week in Nature. This is an example from Pediatric Oncology. Michael Taylor's group from Toronto did a very nice story on childhood cerebellar tumors which mirror conserved fetal transcriptomal programs. And what they did is, first of all, they identified cell types in the developing mouse cerebellum, obtaining data from cells from 9 different time points, embryological but also postnatal time points. And from these data, they reconstructed the cerebellar developmental lineages through pseudotemporal ordering of the cells. From this point of research, they went on to characterize human samples And they did a cell type deconvolution of tumor cell-specific clusters from human medulloblastoma, ependymoma, and pilocytic astrocytoma, single-cell RNA-seq data. And previously, we were not able to tell with cerebellar tumors what is the origin, what is the cell of origin, and at what time this tumor has developed. And by their data, they could nicely show that really the developmental time point is different, and the cell of origin can be identified by this approach because different molecular subgroups of these childhood cerebellar tumors mirror the transcription of cells from distinct temporally restricted cerebellar lineages. So also single-cell analysis help us a lot to understand the pathogenesis of the different cancers. The third aspect is to understand cancer evolution, of course. You have all seen this phylogenetic evolutionary trees where we have a truncal mutation first, then maybe some shared mutations in the tumor, and finally private mutations in some of the tumor clones. And we need to study this in more detail. Here we have the challenge that we need sequential samples, tumor tissue or liquid biopsies. So here the clinicians really have to get hold on the samples of the patients and sometimes follow the patient for a long time to obtain this material. And again, the technique, the major technique so far here is single-cell RNA-seq plus single-cell genotyping. There is a nice example that convinced me a lot from the field of breast cancer, focused on combating subclonal evolution of resistant cancer phenotypes. And the authors of this paper really followed 4 breast cancer patients for a long time. So they obtained samples from the patients here at day 5,586. So you can imagine how long it takes to collect the samples of patients to follow them over time. And they reconstructed the clonal evolution in these 4 patients. And what I find most interesting in this paper is that they were successful in the acquisition of malignant phenotypes after treatment, and they could show that there is an enhanced mesenchymal and growth factor signaling referring to drug resistance, and also decreased antigen presentation and TNF-alpha signaling pointing to immune escape mechanisms. Some phenotypes pre-existed in pre-treatment subclones that then became dominant after chemotherapy. So with these kind of approaches, we can learn a lot about the time development of our cancers. The fourth point, understanding metastasis, maybe with use of liquid biopsies, is also very important. Metastasis is the main cause of cancer mortality. The patients die of metastasis, not necessarily of the primary tumor. But the problem is that we have limited access to metastatic tissue. It's ethically not really appropriate to take multiple samples of every kind of organ in a patient if you can get a diagnosis in a different way. But instead, we can do single-cell analysis of liquid biopsies once we are able to adapt our sensitivity to this approach. So the biomaterial of choice here would be blood or bone marrow, to characterize rare circulating tumor cells or disseminating tumor cells, which are the cell populations with the highest metastatic potential. Again, here serial sampling allows the monitoring of cancer progression over time. In this example, the approach was chosen to profile 77 intact circulating tumor cells of prostate cancer patients that were enriched by microfluidic enrichment, and then the single cell, the pathways enriched in the circulating tumor cells were described, and the patients progressing under therapy, in this case with an androgen receptor inhibitor, compared with untreated cases, demonstrated that there was an activation of non-canonically Wnt signaling. And the proof of concept could be made by the authors that indeed suppression of Wnt5a in drug-resistant cells restored partial sensitivity. So by these approaches, we are learning a lot about resistance pathways coming up under treatment. An even more elegant example is this application of single-cell RNA sequencing to optimize a combinatorial therapeutic strategy in metastatic renal cell carcinoma. So here the authors took biomaterial from the lung metastasis and from the primary tumor. They have grown these in PDX models that we heard about today, and then combined it with a drug screening approach So it's a combination of single-cell RNA-seq of these PDX models plus drug screening, and they could show that cells from metastasis xenografts had a significant different drug sensitivity, signature and sensitivity than cells from the primary tumor xenografts. So probably we have to focus our efforts in the clinic to the metastasis treatment, which is different than focusing it to the primary tumor. The last but one point, understand plasticity. It gets even more complex if we not only focus on genetic heterogeneity, but on epigenetic heterogeneity. So there are different phenotypes. The EMT is only one of it. And although these cells share the same genetic background, epigenetic differences make also a difference in their phenotypical behavior in respect to migration and invasion, therapy resistance, and stem cell-like properties. An example addressing this issue comes from the field of head and neck cancer. So here 18 primary tumors and 5 matching lymph nodes were profiled by single-cell RNA sequencing with a combination of immunohistochemistry to get some spatial information. And here the authors could show that a partial EMT program is present at the leading edge of the tumors, not in the center, but at the leading edge, and that this is regulated by the microenvironment. So there is more computational modeling going on that is linking this feature now to metastasis. The final point, understanding resistance, of course, is something that we are very much interested in the cancer field, and you know that there are different mechanisms how resistance can develop. So first of all, if we miss a primary resistant clone and just focus on the dominant clones, there will be outgrowth of the resistant clone under therapy, and this is a mechanism of primary resistance. Secondary resistance can have many other resistance mechanisms, and we need to learn about both. So here we need sequential human tumor tissue and circulating tumor cells from pre- and post-treatment examples. We need to compare responders to treatment to non-responders, and we need to use different models to learn more about these mechanisms. This is a brief example from our own lab, just a straightforward approach how to apply single-cell technologies after treatment of a targeted treatment, and we see that there are persister cells resisting the treatment, and it is of course interesting to know the difference of these persister cells and the cells that are happily responding to therapy. The final example I would like to share with you is this one. So in this paper, single-cell transcriptomic uncovers distinct molecular signatures of stem cells in chronic myeloid leukemia. And here, resistance, of course, has to do with a fusion gene, BCR-ABL, that all the cancer researchers know. But here, in addition, therapy-resistant cancer stem cells were identified by a single-cell approach, and this gives us an opportunity for the development of better targeted therapies. So, in summary, I hope I could convince you that cancer is a complex but also promising task and an ideal disease to be addressed by the technologies and tools and models developed here in lifetime. I like to see it in a kind of historical perspective. So as oncologists, the first crutch that we got was karyotyping and tumor cytogenetics, helping us to decide on some of the clinical decisions. The next step certainly was extended flow cytometry, bulk sequencing, the Human Genome Project to learn more. Today we have the single-cell approaches and the Human Cell Atlas, even giving us a better prosthesis for our clinical decisions. But if we really want to win the game, we need to further develop our technologies in direction of multi-omic sequencing, spatial 3D sequencing, and for this lifetime, I guess, is the perfect setting to do this. So my final slide, cancer precision medicine tomorrow, what might it look like? So we have the cancer patient, we take the tumor biopsy, but this time we do single-cell multi-omic sequencing. At the same time, we isolate the circulating tumor cells that we also characterize by multi-omic sequencing. We do artificial intelligence. For me, it's still bioinformatics voodoo, forgive me. And then we can identify the cancer stem cells, we can analyze intratumor heterogeneity, we can construct clonal evolution of the tumors, but also we develop the models from the initial biomaterial of the patient. We do PDX models, we do organoid models. While the patient is treated with first-line therapy, we can treat the organoid models as avatars of the patients and learn about resistant subclones and what kind of therapies the patient might respond to. And once we have this information, it might be at the time when the cancer patient comes back with drug-resistant cells, with relapse, and then we might hopefully know from our single-cell studies how to treat this patient better. I started with Rudolf Virchow, I will end with Rudolf Virchow. So he stated, the body is a cell state in which every cell is a citizen. Disease is merely the conflict of the citizens of the state brought about by the action of external forces. So he even envisaged epigenetic phenotypes that are important in the diseases, a brilliant mind truly. And by this I would like to end with this perspective, and I thank you for your attention.
Okay, so we have time for a few questions. And I'll maybe start with the first question. Angelika, so you listen to the talks here and what the many also basic scientists are up to do and what they can do. If you were to say, I would like to have now some kind of interaction with what is developed here, how would you practically like to live that?
So as a clinician, I first need to know what is the perfect material for your study. So I need an SOP that you provide to me, how I should collect the material in the operating theater. Then it will be my task to train my people to obtain this material and to bring it to you in the shortest time frame possible, so that we can then make the most out of this fresh biomaterial. Of course, we would also like to bank some of the biomaterial to use it for more longitudinal sequential studies, and this needs, of course, a lot of time, but is precious material. From our side, we need to make sure that we only focus your efforts on biomaterial with solid clinical data, because if we do it with just some material where there is no solid clinical data around, we can't learn anything. So it should be preferentially patients that are documented in the way we document in clinical trials, preferably patients that are treated in clinical trials. And then I think no matter what the disease is, we can learn the most out of it.
I mean, yeah, okay, so I have further thoughts, but I think over there you were first, actually, and then it's you.
Beautiful presentation. I have a question about the timing in clinical care. The first one is probably a funny observation. So 5,500 days ago, single-cell sequencing was not even existing, yes. And so it's probably about the pace of the technological advancement. How can we keep up with that? The other one is a practical question. What can be accomplished overnight? What can be brought back to the patient within 1 week? And what, since you reported about that, what can be brought back within 21 days?
I think it depends very much on the material. If you can develop technologies that are applicable to routine material, this means paraffin-embedded material in the cancer field, then it's feasible from today because we have this material, and also if it's snap-frozen material, we can come up with the content of our biobanks. As I said, for fresh material, this needs a new setup, and only a few clinicians had the vision that this could be needed in the future, and they already banked live tumor cells, which is not a problem, you just have to think about it. So this material is very precious and very limited, and we have to set it up now to have it at hands pretty soon with cohorts of patients that are meaningful to analyze. It is important to bring it into the heads of the department heads, because really we have so much to do in the clinical part. You have to be behind it and send people directly to the operating theater, because if nobody is standing there grabbing the material from the surgeon's plate, it won't happen. So this really has to be a program that you supervise, and the head of department has to have an interest in it, otherwise it won't work. I think feasibility time-wise, if we have the right material, I think your technologies are so far advanced that we can do it within weeks at least. And of course we would always, as clinicians in the cancer field, start to treat our patients, but first-line treatment is not our problem. In adult oncology we cure approximately 60% of the patients, and pediatric cancer 80% of the patients. So this would always be our first approach with conventional chemotherapy, and for this we don't need the kind of information that single-cell technologies bring. But then when the patient comes back with a relapse, it's much less. The cure rates are less than 10%, and then we need you. But if we start from the biomaterial at the time of diagnosis, we have already learned something in the time where we spent for our conventional treatment, then we have some information at hand once the patient comes back. But we may not rely on the biomaterial of the first diagnosis, because there are some changes between first diagnosis material and then the tumor developing. So the relapsed tumor will have a different molecular composition, and we need to take a new biopsy, but at least some of the information is already there, and we can then be much faster in the clinical situation where we need it.
Yeah, excellent presentation. Over here, this side.
Hi.
Thank you very much. My question goes in regard to the circulating tumor cells, which you're in. So it's not really clear which of these circulating cells really makes the metastasis, right? Do you have any idea how you could challenge the cells and see which one can make the metastasis and has the special program to really be able?
It depends on the field. So some of the cancer fields are quite advanced. Lung cancer, for example, there's a group in the UK working on that, and they can even get PDX models out of circulating tumor cells, which of course is the ultimate proof that something is growing out of these cells. If we just profile them, in my field, in pediatric cancer, there's first evidence that both the circulating tumor cells and the disseminated tumor cells in the bone marrow, for example, are closer to the molecular makeup of the relapsed tumor than compared to the primary tumor. So there is some...
All of them? All of them, or are there different groups?
Also there is a certain level of heterogeneity. Right now we are still fighting with the feasibility to get enough circulating tumor cells to do sequencing with them. So this is the first step. I think once we have solved this step, we can see and look for the heterogeneity in the subpopulations of cells.